Okay, please take your seats, and we're going to move on with the second session, which specifically highlights newborn screening. It gives me great pleasure to introduce the next speaker, the plenary speaker, Olaf Bodamer. Dr. Bodamer got his MD and PhD in Germany, and then did his clinical genetic training at Baylor. He was then the director of the Austrian Newborn Screening Lab for a number of years before coming back to Miami Medical School, where he's now the head of clinical genetics laboratory and a clinical geneticist professor in multiple departments. So without further ado, I look forward to the talk from Dr. Bodamer about newborn screening in the genomic era.
Thank you very much, Hans, for this kind introduction, and I'm very happy to be here, and thank you very much also for the organizers to invite me to talk today about newborn screening in the genomic era. What I really want to focus on, I want to focus on the technical aspects with respect to genomic technologies and as they may apply to newborn screening. But let me start out by making some general comments, some general remarks on newborn screening. I think we all know that newborn screening has come a long way. We are currently celebrating its 50th birthday, and newborn screening was unfortunately not available for this woman who is 52 years old with classic PKU. She shows all the classical features of PKU, and on the other hand, we know the success story in this young gentleman with 3 years of age, also classical PKU, and he's probably meant to lead a normal life. The first hero I wanted to present to you is Robert Guthrie. You see him here depicted with his with an agar plate of his bacterial inhibition test. He was not only instrumental in kind of developing newborn screening, developing the concept of newborn screening, but he was also instrumental to develop the Guthrie cards, and we still use the Guthrie cards for some time to come, and I think the Guthrie cards are not only applicable to the conventional biochemistry methods that currently are in use in the newborn screening laboratories, so they will also play an important role in the genomic era, as I will show you at the end of my talk. Newborn screening, we all are familiar with newborn screening. Newborn screening is defined as the screening of all newborn infants within a geographical region within the first days of life, depending on the region for different conditions, inborn errors of metabolism, endocrinopathies, hemoglobinopathies, cystic fibrosis, and the list could go on and on. What is really important, it's now internationally recognized as an important part of public health for early detection of disorders with a direct benefit for the affected individual, the child, but we also have to keep in mind that there's a family and there's also an indirect benefit for the family and for the public health system at large. We also have to keep in mind that newborn screening is not about the laboratory. Newborn screening has many different components. Here at the center is the infant, obviously, and the parents. But then you have the screening laboratory, the midwives, the birth clinic, the family, as I mentioned. There's long-term, short-term, and long-term follow-up. There are academic centers involved, the public health system, obviously, the Secretary of Health to some extent. And there is close interaction with industry, obviously, and with professional societies. So keep in mind there are different elements, and newborn screening has a different impact on those different elements. There are newborn screening criteria, and I highlighted some of them here. You are all probably familiar with the classical newborn screening criteria, or I should say the classic screening criteria, published in the 60s by Wilson and Jungner. So newborn screening conditions should pose an important public health problem. The natural disease history ideally should be known, and I will show you in a moment that this is still not true for most of the conditions that are currently screened for. There should be an agreed policy on whom to treat. Confirmatory testing obviously has to be readily available. Treatment facilities should be available. And with the aspects of the difficulties we face with respect to reimbursement, we could also argue about that. Obviously, there needs to be a sensitive and specific screening test available that's doable on filter cards. However, having said that, that's the ideal world. What are some of the current challenges newborn screening programs face? There's a lack of evidence about newborn screening, unarguably for most conditions. And I highlighted the publications on the bottom here, and there's a very nice publication actually published in Science by Bridget Wilkin from Australia very recently. The definitions of some of the conditions is not adequately developed, so there's some concern about terminology. The knowledge of the natural history for the most part is very limited, and that may play actually an important role as we continue to expand newborn screening. We have to really make sure that we understand natural history, and there should be a concerted effort in gathering as much information on natural history for potential target disorders. There are still results of uncertain significance, even with expanded newborn screening that is done for the last 10 years or so, and they obviously pose a big challenge. And then different countries use different approaches for adding conditions to the screening panel. I can speak about my own experience in Austria. Austria is fairly laid back. Whenever we had a good idea about what condition to add to the screening panel, we would write a nice letter to the Ministry of Health, followed up by a phone call, and we would get usually approval if we could come up with reasonable arguments. The United States has adopted, which I prefer, a more rigorous approach, which we can also argue whether it's somewhat liberal. And there's also lobbying from different parental organizations, as demonstrated by the addition of Krabbe disease to the New York State program in 2006. So what's the process for nomination of conditions in the United States? If you have your favorite condition, you can actually download the nomination package from this website. You can submit all the information that's available on your favorite condition to the nomination and prioritization work group, and they will review the evidence and the information that's currently available and either disagree and say, well, there's really no need for action, or they approve and they give you a positive vote, which then moves on to the external condition review group. That's an ad hoc review group that will be formed by experts in the field. And then based on this report, there will be a recommendation given to the Secretary of Human Health and Services, which will then either endorse or not endorse the recommendation. There are different conditions, as you all know. This is the table. I think the table is from 2013, mid-2013, which gives you a comprehensive list of the primary core conditions divided into the different types of conditions, so inborn errors of metabolism, hemoglobinopathies, endocrinopathies. There's also cystic fibrosis in there. There's congenital heart disease in there, and there's also severe combined immune deficiency in there, which is not screened for in all states yet. We also have to keep in mind that whenever there are primary core conditions, there are also secondary targets, and this is a complete list of the secondary targets, which are obviously not the primary focus of newborn screening, but they are, if you want to, a byproduct of newborn screening activities. What are additional potential inclusions? We all hear about the lysosomal storage conditions that are currently screened for in several states in the United States, but there are other conditions, there are other lysosomal storage conditions that could be considered, and I'm just throwing out this list without feeling very strongly about either of those. There are certain inborn errors of metabolism, like porphyrias, which are indeed difficult to diagnose, but may have a therapeutic consequence for the affected individual. There are connective tissue conditions like Marfan syndrome, where there are approved therapies. There's hypercholesterolemia, very common condition, which is frequently diagnosed late with significant consequences for the affected individual. There are cancer predisposition syndromes, and I'm talking about conditions that manifest in early childhood, so this could be Li-Fraumeni syndrome, or some of the other conditions that may show symptoms very early in childhood, like FAP, for example. There's also late infantile onset deafness, which may actually be missed by hearing screening. That is significant. And finally, there may be actually pharmacogenetic variants that may be important for medical therapy and that may actually carry a significant clinical utility. So this list could go on, but the issue with those conditions is they lack an appropriate screening test. They may, for the most part, perhaps with the exception of the natural history part, fulfill the criteria I outlined, but they certainly lack, based on the current technologies available, the appropriate and specific and sensitive screening test. When we think about these tests and the technical requirements, what are these requirements for novel analytical platforms? So first of all, the test has to be doable on filter cards, because you don't want to change the screening paradigm of using Guthrie cards for screening. So they have to be applicable to filter cards. They have to fulfill the criterion of high throughput capability, because, for example, in Florida, 1,000 samples are received every day, so you have to have capability of running these large numbers of samples. They also have to have the capability of being able to be multiplexed. You want to run not single tests for individual disorders. Preferably, you would run one test for all the disorders you consider worthwhile being screened for. There has to be an adequate turnaround time at a reasonably low cost. And when we talk about the low cost, that low cost has to be in the dollar, in the single or double-digit range. It cannot be $300, $400 per infant, because otherwise there would be no money left for anything else. It has to be obviously proven high sensitivity and specificity, and it has to be a stable and automated analytical platform. You don't want to have the newborn screening laboratory change the platform every other year because someone has another idea about which platform may be more suitable. We also need to keep in mind, having said that most of the state laboratories that are involved in newborn screening are very familiar with biochemical testing techniques such as mass spectrometry or fluorimetry or plate readers. They are not so familiar with molecular testing. It's just starting with some of the conditions that are screened for, like severe combined immune deficiency, where you screen for DRAG, that's using a molecular approach. So they're just about to enter the molecular era, not the genomic era. What is also important, in order to reduce potential recall and potential false positive rates, there should be second-tier testing available in-house. And now when we flip from using a biochemical test, using a molecular second-tier test to a potential use of genomic technology, you could still have a metabolic test as a second tier to give you additional information of those variants of unknown significance from a functional standpoint. So let me switch gears to whole exome and whole genome sequencing, and let me also kind of give you some general remarks. Whole exome and whole genome sequencing. When we talk about whole exome, we have to keep in mind we sequence only about 1% of the genome. So 99% of the genome is actually missed, but whole exome will entail analysis of all the coding regions. You always have to keep in mind that the capture rate has to be as close as possible to 100%, so you want to capture all the coding regions. In reality, it's not close to 100%, it's more in the 90 to 95% range, which means that 5% of the coding regions will be missed. That's due to technical reasons, due to the GC richness of some of the genes. Mostly the first exons are affected. What is a big advantage? It's unbiased. You don't need to have an idea about your phenotype. You don't do targeted gene analysis. You do an unbiased analysis, and it does not require prior knowledge of candidate genes. It's expensive, obviously, compared to current newborn screening by several fold. but I will show you that there may be some approaches to actually reduce cost in the future. Whole exome is by all means faster than whole genome analysis, and it's easier and simpler to analyze, as I will show you in a moment. Can be done in different populations, and it will identify any rare variant, and I listed some of the variants here at the single base nucleotide level. And its diagnostic yield, if we use it in pre-selected populations, is about 25 to 30%. What are some of the challenges? In particular for whole exome, we have to understand its limitations. It does not cover 99% of the genome, including, I mean, these are mostly non-coding introns, but there are some regulatory elements in the genome that may not be analyzed. I already mentioned that capture is not 100%, so we're missing out on some of the coding regions. We also have to keep in mind that the read depth, that's the number of parallel reads, is variable, and you would prefer to have as high as possible read depth, at least 30x. So in order to analyze your region of interest at a sufficient sensitivity and specificity, you would like to have that target region at least sequenced 30 times in parallel. And that may also vary significantly from platform to platform and from laboratory to laboratory. The biggest challenge I foresee is the bioinformatics. The generation of data is huge. Filtering out the variants you are looking for is a big challenge, and I will give you some examples. And there's also limitation, as this technique will not detect triplet repeat changes, for example, larger insertion, deletions, complex rearrangements, and splice site variants may be challenging. It can be addressed with a sufficient bioinformatics pipeline, but it remains a challenge and it remains a limitation. We also have to be aware of the fact that on average we will identify 5,000 to 10,000 variants per individual. And the variants of unknown significance are obviously, in all the papers you read these days about genomic technologies, these are the biggest challenge, because you will certainly find variants of unknown significance where you need a second test, you need a functional assay, or you need a biochemical assay. If we take PKU as an example, if you would identify a variant of unknown significance in the phenylalanine hydroxylase gene, you can follow up with measuring phenylalanine, for example, in that individual. So you would need a second test to really interrogate those variants. How many variants do we expect? There are several publications. This was a very nice publication published in 2013 by Dorschner and colleagues, who identified in a genome project 3.4% incidental pathogenic mutations in dominant genes, so for example BRCA1 or FAP, in individuals of European descent, and about 1% in African Americans. And these were purely incidental findings. The Baylor published a study in 2013 based on their clinical exome experience, and they identified in each of the 250 patients that were included in that publication, on average about 300,000 different nucleotide variants and small insertion deletions, and they identified still 300 to 600 potentially pathogenic variants in each patient, and ultimately they had a diagnostic yield of about 20%. In our own experience, we sequenced about 31 patients, 30 patients with including family members, and we found on average about 300 potentially pathogenic variants per individual with a diagnostic yield of about 30%. So there's still a big gap. We could argue these are obviously clinical cases, why this gap is, but there are different challenges that still need to be overcome because prior to us even considering newborn screening, using these technologies for newborn screening. What does the workflow look like? Obviously, you need to prep your sample, and you already see in this picture here that this needs to be done in a high throughput manner using a high throughput platform. We'll show you some details in a moment because you want to extract 1,000 samples per day, so this has to be done in a high throughput manner. You prepare your library, you enrich your target sequences. followed by cluster generation. So you generate clusters of your targets of interest, followed by sequencing, and then what looks fairly simple here, bioinformatics and sample storage. But believe me, bioinformatics is very complex. This is a simplified algorithm that's currently in use at our institution. It starts out with extraction steps. Then you have a library prep and sequencing step, which is kind of this middle section here. Don't want to go through all the details. But then the most important part is actually the lower part here, where you have to go through a series of filter steps to identify your variants. This is obviously done with the use of different publicly available databases, such as the dbSNP database. You want to filter out any common variants. We have access to the 1000 genome database to really make sense of the variants, obviously in the context, in the clinical arena, in the context of the phenotype of the individual patient. This will be a challenge for newborn screening because you don't have a phenotype. Yes, you know what genes you are looking for, but this may be a big challenge because there's no phenotype you could potentially correlate it with. You could obviously ascertain the genes of interest and only look at those genes using potentially not an entire exome, but a more sophisticated gene panel. We also have an automated bioinformatics system in-house, and I would ask you to write down this website because this is free of use. It's a web-based application. If you have next-gen applications, you can actually download the sequences under your name, under your login name, using this software. There's potential for worldwide collaboration. We have about 180 worldwide collaborators in about 23 different countries sharing data. This is obviously more applicable at the moment for clinical cases when you identify a gene of interest and the gene has not been associated with your clinical phenotype. This tool may be actually able to give you a nice idea on what other groups might be interested in that particular gene and what other institutions may have found similar variants. We currently have in excess of, I believe, 1,200 exomes and about 600 genomes uploaded into that web-based bioinformatics pipeline. So the workflow, and let me just speculate, the workflow for genomic newborn screening could look something like that, where you have obviously high throughput DNA extraction, your library prep and capture, your sequencing, your bioinformatics and reporting, which is obviously automated based on the genes of interest, and then your second-tier testing, which is either functional or metabolic. The DNA extraction can be relatively easily automated, which we have done using two different systems, a Siemens tissue preparation system that allows DNA extraction from 96 well plates in several hours, and we also use the QIAGEN Symphony system, and you see some of the examples of the extraction here. You get very good yield, high quality DNA extracted from dried blood spots. What are the different systems we looked at? This is an incomplete list of the different systems. You see there are different levels of automation here, different levels of throughput, and obviously different levels of cost. There's one nice protocol that's used by New York State for Krabbe screening, which is a manual or semi-automated homebrew protocol at a very low cost, has been published in 2013 for anyone who is interested. We have compared the Siemens system and the QIAGEN system in a number of individuals, and this is just to give you an example of what the yields would look like. So if you use a single 3 millimeter punch, on average you get a DNA yield of 60 nanograms. Conventionally, you would need to run a whole exome, you would need 3 micrograms. So we are really kind of going into the low DNA input arena. You see, if you use 3 punches, you get about a 3-fold increase, and it's fairly comparable between the different systems. What sequencers are available, that's I think also important if you consider, and I think the cost aspect here and the high throughput aspect is very important, and this is taken from Dr. Kedouche publication in 2013 comparing the different generations. So we talk about first generation when we think about capillary sequencer, second generation, and third generation, and fourth generation, and you see the different costs with respect to instrument, different costs with respect to price per genome, time per genome, and then the accuracy obviously plays an important role because there may be some issues with respect to sensitivity and specificity and accuracy using these different platforms. And the price has come a long way. When you think about the first human genome completion cost in excess of $10 million. Now Illumina just announced the $1,000 genome analysis. So we have come a long way, but we are nowhere near the cost we would need for newborn screening when we talk about $15. So I think there's another generation that needs to be, cycle that needs to be completed if we even think about using genome technologies for newborn screening. When we compare the different second generation sequencer, again, you can look at the table with respect to throughput, instrument cost, time to result, which is important, and there are several, if you look at the time to result, for example, it's very doable for newborn screening, error potential, and the cost per gigabyte here, again, taken from the same publication. I want to highlight one fourth generation sequencer, and I don't have any relationship with that company. That's GnuBio. GnuBio is a very interesting approach. GnuBio is a startup company that was initially started, I think, 4 years ago as a spin-off of Harvard, has been recently acquired by Roche. And that's a sequencing technology that's based on microfluidics. You see that there is a PCR primer library, and these are little droplets that go through these capillaries. The genomic DNA is introduced at this point here. Then the amplification and enrichment steps, they take place in these hundreds of loops of these microcapillaries. And then you have a sequencing probe library based either on an entire exome or based on your specific panel. And there's an automated readout and a detector. It's very nice because this instrument, according to the company's website, is about $50,000. It's currently in beta testing. It's the size of a desktop printer. The sequence panels are done in cartridges, so you just load apparently the cartridge into the instrument. And this is a comparison comparing sequencing of 50 genes using a conventional next generation sequencer, and you see the time that's used and the different steps with the microfluidics approach by GnuBio. It's a significant reduction in time. And also when you look at the cost, I talked to some people there, when you have a cartridge that is able to sequence 1,200 genes of your interest, that cartridge is in the $200 to $300 range. The big disadvantage of this instrument, it's currently only made for single use. So you have to pipette every single sample into the sample port. So it's not amenable to high throughput applicabilities, but it will be, I think this is one way where the future could go, that you have these desktop sequencer that may have multiple ports or be able for high throughput sequencing. Certainly from a cost and accuracy standpoint, what I have seen by talking to them, this is certainly one possibility to go. And like I said, I don't have any involvement with that company. So I mentioned the time aspect, and there have been a couple efforts by the Kingsmore Group in Kansas City, and this is a very nice publication that has caused a lot of publicity that was published in 2012. And this is the workflow, and you see the time aspects here. It's very doable, and within 50 hours, you have a verbal interim report of the diagnosis, obviously pending confirmation. So this is for single selected cases. Dr. Kingsmore published cases from his neonatal intensive care. This is certainly a nice and a doable approach that's feasible. And this is also important in the same publication. They published the first 9 cases. They are listed here. And what is also important to understand, we are not only sequencing the nuclear genome, we also obtain information on the mitochondrial genome. So some of the mitochondrial conditions will be diagnosed as well. And you can go through the table and you see listed the number of variants that have been identified, which is a staggering number. These columns here, you see when you start classifying those variants as to likely benign, likely pathogenic, using the ACMG criteria, you see that there's, using their filter pipeline, there's a significant reduction. And when you bring in your phenotype, you further reduce your number of potential variants explaining the phenotype to a reasonable number that's certainly, from a clinical standpoint, manageable. But again, when you look to the far right here, you see that some of these cases actually go undiagnosed, again, reflecting the diagnostic yield of about 25 to 30%. What this group also has done, what I really like, is they actually compared different target enrichment methods, different sequencing methods with each other, and they came to the conclusion here to the far right that there's actually very good agreement in the identification of the different genotypes. So I think that's also important when we talk about new technologies as they enter the newborn screening laboratories, that there has to be due diligence in kind of really comparing different platforms, identifying the gaps and the challenges of different platforms, and certainly putting some data together with respect to specificity and sensitivity. So we have started our own little program looking at these technologies using de-identified dried blood spots. So these samples were provided from our referral center, including some of the current core conditions. We have a low DNA input protocol that actually allows us to use 50 nanograms of DNA. LibrePrep is mentioned here. Initially, we used the HiSeq 2000, and we are hopefully switching at some point to the HiSeq 2500. And if QNU-Bio really demonstrates the potential use and the applicability of its system, we hope at some point in the future to also use their system for comparison. We use our in-house bioinformatics tool, the GemMap, and the goal obviously is to achieve 100% sensitivity and greater than 99.9% specificity with a reasonably short turnaround time and high throughput capabilities to run 500 samples per day. I think I got carried away when I wrote this because that will not be doable probably before I retire. So the low DNA input protocol, anybody who is interested, we are happy to share this protocol. This is actually very doable on 50 nanograms DNA, high quality DNA. You see that we get reproducible peaks at about 300 nanograms. So that's very good, very robust protocol. Easily done on dried blood spots as shown here. Here you see 3 examples I just picked randomly. So here we had 3 punches and a single punch, and you see with the variable sample input, we were able to get good yields post-capture using post-capture PCR. So this pilot project basically has started. We're still looking for funding. So if you know of any donors that is willing, just see me after that talk. So the first set of 10 samples included 6 supposedly healthy individuals and 4 with a defined monogenic condition, 2 of them with mucopolysaccharidosis, 3 actually of them with mucopolysaccharidosis, 2 type 1, 1 type 4A, and 1 Fabry. And we were able to identify correctly to call all the genotypes and did not identify any pathogenic variants in the 6 controls. However, having said that, that was a big challenge because on average we had about 5,000 variants per individual with about 30 to 50 predicted pathogenic variants identified in each individual, mostly heterozygous variants in recessive genes. And that really has to be really thought about and has to be laid out very nicely, because otherwise you may end up in a mess if you report out all these potential pathogenic variants in a supposedly healthy individual. And the analysis is still ongoing. So what are let me come back to the technical requirements. So genome technology is definitely, as I've shown you, doable on filter cards. It still lags and will lag for some considerable time the high throughput capability. It certainly has the capability to multiplex. You can run multiple genes in one analytical run. Adequate turnaround time, as Dr. Kingsmore has shown, is doable. It certainly has a higher sensitivity and specificity. Whether it has the sensitivity and specificity that's required for newborn screening needs to be seen, because you really need to test all the different genotypes. And it's a stable automated analytical platform. But knowing these systems rapidly change, so I think they are not quite there yet to provide a stable platform that will not be changed for the years to come. And low cost per sample analysis, obviously that's not the case. So let me summarize. So newborn screening, even expanded newborn screening, our current programs still face many different challenges. The genomic technologies, in my view, are not ready yet. There's a lack of scalability and high throughput capability. The cost per sample is obviously prohibitive. Turnaround time, I put in brackets here because I think that's doable. And there need to be carefully planned studies to address the accuracy, the sensitivity, and the specificity for your target disorders of interest. So maybe one way to start would be, instead of starting with the whole exome or whole genome, to start with a gene panel of your particular interest. And obviously, I didn't even touch on the ethnic, social, legal implication. They obviously need to be addressed prior to the initiation of such studies. There has been some initiatives by GHRI. They funded 4 different projects. You don't need to read through that. If you Google, you will find all the details. One has been funded, one grant application has been funded at Boston Children's. They essentially do genome and exome sequencing on infants in their NICU, and then present those genome results and the newborn screening results from the newborn screening program to the parents, and obviously question the parents about their views on the use of genome technologies, and they will also obviously compare the different diagnostic yields between the two. The second study was given to Dr. Kingsmore, who I mentioned already. This is kind of a study that's focused on more a clinical use of whole exome and whole genome technologies in the setting of neonatal intensive care to improve turnaround time, to streamline the different analytical protocols. A third study has been given to the University of California in San Francisco. They use a slightly different approach. They actually screen for common conditions or conditions that are currently included in the newborn screening panels and compare genome and conventional newborn screening, and also obviously assess the different views of the parents. And the study that was approved by the funds that were given to the University of North Carolina at Chapel Hill uses a similar approach by interrogating conventional conditions that are included in newborn screening using the different technologies. So there's a big push from the NIH to fund those studies and to move forward with genome technology, but really a word of caution. I'm not sure whether the future will look like that. At the moment, it looks like we're moving into that direction, that you sequence once, you have whole genome sequence data, you have a continuous medical annotation, obviously with implications coming from new research that will also provide education algorithms to practitioners. The individual patient, or the individual, I should say, will have a personalized genomic medical record that could interrogate with the electronic medical record. I'm not endorsing this, I'm just throwing this out as food for thought. With that, I'd like to close. There's another hero of mine, Rodney Howell, who really has been instrumental in expanding newborn screening and who has been really very helpful in moving the field forward. And with that, I'd like to close. And thank you very much for your attention.
Oh boy. So that was brilliant, beautiful description of the technological piece of newborn screening. And so, wearing my clinician hat, I wonder how we will manage to convey the information from whole genome to the probably 30% or more of all live births. That's obviously a paradigm shift that all physicians couldn't even do.
Right.
So how do you imagine this could occur, or would you endorse, obviously, then just going from a panel of genes using next generation sequencing and scaling up as the resources allowed?
Right. You know, I think we are on the same page here. I think I'm very concerned about use of genome technology for newborn screening at this particular moment, because like you said, everybody would be overwhelmed. I still remember the days when newborn screening expanded in Austria. I, as a clinician, was overwhelmed, despite the fact that we expanded from 6 conditions to 12 conditions. So can you imagine expanding from 34 conditions to everything? I think this will not be doable, and I think this should also not be the way forward. I think there should be really a period where there is a translation of use or a comparison of the use of current technologies using this particular core set of conditions, using genome technology like a expanded panel, gene panel, using whole exome capture, just focusing on those. Then bringing in additional conditions that may fulfill certain criteria, and I think I briefly mentioned the criteria. The criteria really have to be redefined and revisited, because I think some of them are obsolete and some of them are not really up to the current knowledge. But I absolutely agree. I would not jump from 0 to whole genome, and I didn't want to imply that.
Good, thank you. I just want to mention to everyone that tomorrow at 10:15, Dr. Saunders, who you referenced, will be speaking about similar topics.
Right.
Other questions? No questions on this topic.
It's kind of concerning.